In this video, we will discuss about what are gram positive and gram negative bacteria. Gram positive and gram negative, and why they are important to study. So, gram positive bacteria, let us understand this first. Gram positive bacteria has some plasma membrane. After that, they have a thick cell wall. After that, there is another layer that is called as capsule. And whereas this is the plasma membrane. Plasma membrane, cell wall, thick cell wall, and this is capsule. And how this gram negative bacteria look like? It has this plasma membrane also, similar. It has the thin cell wall. Cell wall is not that big. After that, it has another plasma membrane, and then it has this final layer that is capsule. So what happens is that since there are two plasma membrane in gram negative bacteria, they are difficult to affect. They are difficult to get affected by antibiotics, whereas since it has only one plasma membrane, it is easier for the antibiotics to affect this gram positive bacteria. So whenever someone is dealing with bacteria, in order to identify the correct steps and correct steps for the treatment or for the analysis, they need to first determine whether the bacteria is gram positive or gram negative. So based on that, they can decide their subsequent steps. So how they decide whether the bacteria is gram positive or gram negative? There is a test called gram stain. Gram stain test. And why these are called gram positive, gram negative? Because it is discovered by the scientist whose name is Gram. So what happens in gram stain is this is a 4-step process. Initially, they treat the bacteria with crystal violet dye. So what happens is crystal violet dye will pass through all this capsule, cell wall, plasma membrane, and settle inside the cell. And same happens in this case. After that, they treat the cell with iodine. Iodine will also pass through and make a bond with crystal violet. And same thing happens in this case. The next step creates a difference. After that, they will wash it with alcohol. What happens when it is washed with alcohol is that the capsule in this case, in the gram positive bacteria, the capsule gets washed away. Capsule gets washed away. And this also cell wall gets shrinked. So now the cell wall gets shrinked, and capsule is no longer. And whereas in this gram negative bacteria, what happens is this capsule get washed away, and this plasma membrane also get removed. Plasma membrane also get removed. And after that, this cell wall here also cell wall gets thin when they are washed with alcohol. And since the thickness of the cell wall is high, this molecule, this crystal violet bonded with iodine, will not be able to move out, whereas in this case, gram negative bacteria, crystal violet with iodine bond will be able to escape the cell. So it is no longer there in the gram violet at this point. So at this point, what we can see in the experiment that gram negatives are transparent and gram positive acquire a purple color. These bonds gives a purple color. However, in order to also visualize this gram negative bacteria, we add another dye, that is safranin. What safranin does is it will then go inside and bonds and stick at the plasma membrane. And this safranin will give a light pink color. So this will show a light pink color, and this will show a purple color. So it here also light pink color is the characteristic of this gram positive. However, in the competitive environment of purple and light pink color, purple being the more contrast color, it will appear in the experiment, whereas in case of gram negative bacteria, it is competing with the transparent. In that case, the light pink is more prominent or more competitive. So we will see gram negative bacteria will reflect light pink, whereas gram positive bacteria will reflect purple. So in this way, people are able to identify whether the bacteria is gram positive and gram negative, and after that they can decide what are the subsequent steps they need to take in order to treat or analyze the bacteria. Thank you.